it's
You don't
literally
think
one page.
to explain it though?
It doesn't take that long.
You don't think so?
No,
Over
it
ATP
doesn't.
and how
We're
it's
not going
hard.
over ATP, we're just gonna mention ATP.
Okay, so
ATP
So
is
not
the
going
next
over?
unit,
Okay.
ATP is the next unit.
here's my kind of goal for us. I think what we should do today, and when we get Ms. Barker here and Mr. Mahoney here, I think we should talk about how we're gonna put in our timeline, how we're gonna fit unit 3, which is, well, depends on how you look at it. Unit 2, which is homeostasis and cell transport, how we're gonna finish that up. And then where do we put, how do we do photosynthesis and cell respiration? And then how do we fit in cell division, unit 4, unit 5, unit 4, how do we fit in cell photosynthesis, cell respiration before the benchmark, which is on the 8th. I really wish we could give the benchmark on the 9th and the 10th. That gives us a little more time, but the district calendar has us doing a bridge day here and here. Now, this day is not an early release, is it?
No.
Okay, so we don't have an early release till Christmas,
No.
right?
Right, we don't have 2 early releases.
And then we've got, in here, we've got a PD day on the 20th, and then we've got fall break that falls in there.
Okay,
But that's
here's
after the
my
break.
concern. We've got the role of nucleic acid in protein synthesis, and then we have gene expression, and then we have genetics.
Right.
We are gonna spend,
We're gonna spend 19 days on gene expression.
and the kids are not going to
We're gonna
be.
spend 4 weeks on genetics.
We're
We're
gonna
spending
spend,
9
we're literally
weeks.
spending almost an entire 9 weeks with
And
role of
after
nucleic acids,
Christmas.
protein synthesis, gene expression, and genetics. The way the calendar is written,
But then we're also going to January 16th with genetics.
and we're still going, yep.
I feel like that's a wee bit
We,
much.
remember, we did this last year, and we had way too much time in there.
Yeah, we had way too much time, we didn't have enough stuff to do, and that's why I'm suggesting.
And we did like 2 projects.
Mitosis for after fall
This
break.
needs to, mitosis needs to come after,
Is it
fall
gonna be
break.
on the benchmark?
So what I'm gonna suggest, since, and when Ms. Barker gets here, we can talk about this. I'm gonna send Sandra a message, and I'm gonna say, we are just jamming, I'm gonna send the district, specialist the message and say, we're just jamming way too much together. We need to spread some of this out. We need more time with photosynthesis and respiration. We need more time with cell division. We can't just jam all this together, just to try to get it all in for the end of the first 9
And
weeks.
then we're not making it fun for the kids.
Because gene expression and genetics, 19 days. 19 plus 20 plus 12, that's what, 31 days?
Yeah,
31 days for,
it's 6 weeks.
that's a whole 9 weeks
Yeah.
that we're
9
spending
weeks.
on quote unquote genetic
No,
stuff.
I can understand, I mean, genetics was a long unit, we spent a month on it,
Right.
okay.
But that was with 2 projects in there, we basically just kind of threw 6 days, just whatever.
So I think we need to work on our timeline. We need to look at, I pulled some questions, and if we get a projector, we're gonna look at some of last year's EOC questions, because I pulled those from, not Lead Forward, but I went back to the STAR test itself, and I pulled those, because Lead Forward said they didn't test cell transport last year, but in fact, they did test cell transport. So we need to look at that. And then we need to think about scaffolding. How are we gonna scaffold back in biomolecules and this
Well-
information from biomolecules into this unit, because it does come back.
Right,
We need to talk about enzymes again, and we need to talk about those biomolecules as they relate to functioning in the cell. Phospholipids, proteins, like transport proteins, channel proteins, peripheral proteins, glycoproteins, glycolipids.
Sure.
How we're gonna scaffold some of that back in. And then I think we ought to, by the time we end today, we should have some SMART goals kind of set for where we want to be and where we want the kids to perform at. That's kind of the goal. If you look at our EOC from last year, just kind of jump in here, we hit 96%. I went back and looked, and the questions that the kids really kind of got tripped up on was some of the cell transport kind of tripped them up, and I think it may be a function of the way the questions were written and the way the questions were posed to them, versus did the kids really understand it. We spend a whole lot of time on, hypertonic, hypotonic, isotonic, and
how many questions are on there?
there's none. They didn't
No.
ever test those terms. But they
that
do
doesn't
test
mean
them.
they won't test
They
it.
tested like the picture of the membrane with a protein embedded in it, and they had to identify the structure, the protein, and what is the function of the protein, and what processes is that protein aiding the cell in, or what processes are going on there, and it was almost maintaining homeostasis, transporting molecules.
Yep.
So we need to kind of think about the way that they tested it last year, and how's that gonna affect how we're gonna present that information to kids. I think our greatest need this morning, is probably digging into this timeline, and our scope and sequence and our calendar. That's probably where we're gonna spend most of our time, is kind of fleshing this out, because we're short on time, and not necessarily short on time, but we feel like the district has got us in a, kind of a rush. I think we're gonna hit timeline first, and then we'll look at some questions.
So, do you think the progress check is gonna get pushed back?
I think the progress check, I'm gonna go ahead and, email Sandra today and tell her that I really do think that I don't feel any other campuses are ready for this, but we're not gonna be ready with cell transport, and we're not gonna be ready with, photosynthesis, cell respiration.
And we weren't last
And
year.
even to that end,
Because that means the benchmark's pretty useless.
even to that end, just giving 4 days, 5 days, 6 days, 7 days.
It's not 7, we have to review.
We gotta review, so 6 days for mitosis and cell division and cell differentiation.
Yeah. It's not
That's
going
gonna be a jam also.
Yeah.
And I'm trying to figure out, I know I don't think we've ever done cell division and, differentiation in the first 9 weeks. I think we've always done it in the second 9 weeks.
Last year, we did part of it before
Before we
the
went
fall
on,
break.
before we went on fall break.
And then we had the
But-
benchmark, we did it after,
Okay.
or was that spring break? did
We
some-
split some of it during spring break. I can't remember
I
what
don't
we did.
I don't think we split it up.
I can't remember how we did it last year. I have to go back and look at my calendar to see for sure. I just thought, I think she jammed it, I think we did it after the fall break, because I don't remember doing it before. Fall
I
break.
think
How
that-
she's arranging her thing
I've
is
got
how
it on
Savas
this.
has it arranged.
Well,
And
it's not,
it's too much in
And that's-
too little
well,
time.
think about it, 31 days in the second 9 weeks.
Our first
And we're
progress
gonna
check
do-
was December 10th last year.
Our first progress check?
Was December 10th. That's what it says right here in the folder from last year.
Our first probably the benchmark.
Look at the benchmark there. That's a month early then.
Yeah.
Yeah,
Well, she's giving, we're only doing
Progress check.
one
number
benchmark per 9 weeks, and we're
Yeah,
doing
I have
one
all
progress
that we
check
did
per 9
last year
weeks.
right here.
Last year we did 2 progress checks each 9 weeks,
Okay,
and the
but-
benchmark, because we had 3
But
summative.
the-
I'm looking what's on it, cause I see prokaryotes, eukaryotes, identifying the bacteria cell,
But the first
differences between eukaryotes and prokaryotes,
9 weeks were a wash for us because.
Yeah, we had to start over.
talking about
We did the
enzymes,
reset.
enzyme denaturing, what's the role of enzymes?
About what time did we talk about those?
I wasn't teaching it last year at this time, so.
Oh, yeah, you got shunted, like.
So if we're looking, and I went back and like I said, I went back and looked at those EOC questions from last year. The questions that tripped the kids up was the cell transport question about the cell membrane and the function of the transport proteins. Being able to identify the transport proteins and then say what it does. The other one that tripped them up was the photosynthesis and respiration, and I don't know why that trips them up every year, but last year's question was a table that they had 4 tables, and each table showed the inputs and the outputs for photosynthesis and for respiration. And I don't know why they missed that, because to me, it looked like a pretty obvious question, but I think the kids get lost in the sauce, and they kinda can't see the forest for the trees. We need to get them to grasp the big idea. What is the end result of photosynthesis? What is the end result of cellular respiration? And how do those two interact? How are they related to each other? I think that's the big idea that we want to get. So looking at last year's data, actually the kids most of the kids did fairly well on that photosynthesis question, but the cell transport question is the one that tripped them up
Well, on
the most.
the benchmark from last year, there's only one tonicity question, one.
Yeah, there's only one tonicity question, and that was, can you tell me what that was? How did it read?
It was.
Was it a hypertonic, hypotonic, isotonic question?
One
Yeah,
second, I
she's
just
just
saw.
literally, she is doing what we did last year, cramming it all in again, so basically the first 9 weeks is gonna be a wash.
Yeah.
Because that's exactly what she.
So-
Here it is.
We're gonna have.
Researcher
a
wants to
lot.
see how blood cells respond to a change in tonicity. The image shows a red blood cell floating in a saltwater solution. The particles are the solute, some of which are inside the cell. Choose the option that identifies the missing terms. It says the solution in the picture is blank and the cell is blank homeostasis.
I think what she's doing is she's using that Savas calendar,
Yeah,
and if you went through Savas and you use Savas with fidelity, it would take you that many days to get, because Savas breaks genetics up into 2 chapters. And so if you add the roles of nucleic acids in there too, then it breaks it up into 3 chapters. But I don't think we need 31 days to cover to cover roles of nucleic acids in protein synthesis or gene expression. I think those, because
Well,
role of nucleic acids in protein synthesis, that's 12 days, folks.
yeah.
All we're teaching them is what does DNA do, what does mRNA do, what does tRNA do, what does rRNA do.
and. we're
That's
gonna
basically
do
it, and if you can't do, if they can't get that in 12 days, something's wrong. That's a 3-day lesson, at most,
Yeah.
at most. And then she's given us 9 days for gene expression. Well, that's just protein synthesis, that's turning DNA into RNA, RNA into proteins and building the protein. And she's given us 9 days for that, so that's 21 days. And we used to do that in a week or a week and a half. So I think, I honestly think we need to I'm gonna send her a message, and I'm gonna suggest that we move that progress check, or, I don't know.
I don't think she's gonna move the
Not
price.
move it, but let's just,
Change what's on it?
if we can move this to here,
Yeah. Then the benchmark has to go away.
and then the benchmark, take off the benchmark, take off the cell division and the differentiation, and just make it biomolecule cells and cell processes.
Okay.
Let me see what's on the second benchmark.
Because the kids can do that.
Second part. of it.
How many days should we have for mitosis?
That's something we're gonna have to work out. We need to sit down and think about it.
But honestly, if we don't have to do another notebook setup between now and fall break, I'd be pretty happy with that.
I would be too.
Cause we are legit cramming 2 units.
I mean, even the second
No, we're
progress
doing
check
like 3 units.
is this, almost the exact same information from the first one.
So if we look at our calendar, let's just go ahead and see, how will we finish up,
Oh, yeah,
how will we finish up cell transport homeostasis?
Are we keeping the progress check?
Leave the progress check here.
Okay,
So we've reviewed the day before.
yep. And if I don't have anything to, like, make the book it with, does anyone have, like, you just want me to do old questions? You want me to do?
Mark sent her the CUAs.
Yeah. I mean, we can just
She
use
may
the
be
one
able
from
to
last year.
copy
It's the
them.
Okay.
same.
Yeah.
Okay, I can send you the old
We could
CUAs
just
for that.
give those, the old CUAs.
What about the one that
and-
you used last year?
Okay.
Yeah.
So tomorrow, are we all gonna hit active
I
transport
didn't
tomorrow?
have one last year
That's what I'm
because we were
gonna
doing SATs.
I won't be here because of the Bengalti and stuff, but that's what I was gonna do.
Oh, we
with
didn't do,
the
oh,
notes
okay,
wise.
first night we.
I doubt I'll need the whole class.
Yeah, I
for
did
each
that
day.
yesterday.
So
Oh, you did,
that's
yeah,
it,
that's okay.
Yeah, I did active transport.
I was gonna do active and passive, whatever, I don't know how much
Well,
that is.
We've
Passes
got-
a lot though.
We've got 2
But I'll-
pages on active,
Doing
and then
his
we've
notes
got three pages on active.
In
And
the
then as soon as I get
exercise.
done with active, I'm moving straight on to passive. So I'll probably finish passive on Thursday.
Yeah, I
Cause
have
active's not gonna take a whole day.
So you're gonna start with passive here too?
Not
big
yet.
question mark,
Already even. Today,
because there's a lot of
we're gonna do
activities
passive.
for the kids
Yeah.
to do with
There's
them.
lots of activities, so.
Yeah.
So if we started with passive transport, we got through active transport tomorrow, then we introduced passive transport,
Yeah, and it goes farther.
and then Thursday,
Finish passive.
Finish up.
finish
the-
passive.
Alright, what about the egg lab, we'll do that over the weekend?
And then we can do this like a warm up on Monday.
So Friday, set up egg lab.
Yeah, but
That's
we
a
gotta
good idea.
we gotta get the shells off.
And then we can do
So
a
we
warm
said,
up.
if we put them in vinegar on for Friday,
Code yellow, what does that mean?
Oh, shit.
you're gonna put them on,
What?
put the eggs in vinegar on
This is
Friday.
tomorrow, though.
And then Monday,
Tomorrow.
Monday,
code
put 'em in the
yellow
different
is we're
solutions.
doing the- It's the,
and then let them sit all week, and so
testing.
the kids can see them.
Just
We
that.
could,
We go to
Just this.
the auditorium,
Did you have the
and
drug test?
so.
No.
We take the kids.
We go, they give us a list of kids and we go pick them up out of the rooms.
Well, we're not.
He'll be babysitting in the,
Maybe we can use it as a break.
Well, I won't be there.
Are you leaving early tomorrow for the
The
game?
whole day.
So what's the plan for Friday then?
Yeah, well, that's what so back to track, what activities are we talking about here?
So we've got page number.
Gummy bear!
No, we've got page number 39, which has diffusion. We've got page 40 with osmosis and cells.
What's the activity?
Should've
then?
come
It's
clear.
this.
You're not good.
Okay.
Diffusion, osmosis, what else? Do we have,
We have this page.
then we have the tonicity in the water, okay, osmosis.
I'm not gonna spend a lot of time on Tennessee. It doesn't seem like it matters that much for.
And what's osmotic regulation? I couldn't even say it.
Water
Osmo
regulation.
Oh.
How much water you got in
It's
the blood?
water regulation in and of itself.
I was like, man, I haven't heard that word.
So
before.
Friday, what are we looking at?
Do you wanna do the gummy bear lab on Friday, or do you wanna nix the lab?
Well, because let's see if we can fit in. We're keeping the progress check. We still have to get to cell respiration and photosynthesis. How much time do we need for that?
Gummy bear lab takes all period.
But the gummy bear lab, all they have to do is measure it and possibly, what did they weigh it?
How much time are we gonna take for the notes?
Let me look at one thing real quick. I don't know if you've been using the TEAS guide. It's teatsguide.org.
Is that the one that?
That's where all the specificity and everything it used to be called something else, and I struggle to remember what it used to be called. But if you go
We
there,
could do the muscle fatigue lab on Friday.
We still gotta get through photosynthesis and cell respiration.
No,
No?
not with the muscle fatigue, that's how we intro it.
Okay, okay,
Because
it's a good
you gotta
intro.
talk about fermentation, because that's what's gonna be.
But you
on-
will
at the
fermentation
beginning
start
of
this,
the-
though?
No, but it's the beginning
Okay.
of cellular respiration.
so 11A is on the test.
Anaerobic respiration, but yeah.
Does say 11A,
Because you have to
11B.
break up with the glycolysis outside the mitochondria.
Explain how matter is conserved and energy is transferred during photosynthesis cell respiration using models, including chemical equations for these processes. Okay. So it literally,
Fermentation lab's also
I want to
fun.
see here,
Use
photosynthesis.
chemicals, fun.
Chemical process by which plants and I don't think,
I'm gonna have 100 grades for this quarter.
I already
That's
have,
fine.
I already have 8.
7 mL by
I already have almost enough to submit for, like,
report
report cards.
Yeah, that's pretty
Release
well.
waste product, okay. It does not say here. Students should understand that when matter is changed through various cycles, it is accompanied by a transfer of energy. Energy enters the cell through the process of photosynthesis as light energy from the sun is transformed to chemical energy, and chemical respiration, cellular respiration, the glucose is
Why do you say-
ingested
we
by
don't
an
do
organism, is
those
broken
activities?
down, and the energy transfer,
I think-
carrier molecule
We
ATP.
need
do?
to, I think so, Thursday.
I mean,
Passive transport and the
Yeah.
infusion one. The tenacity one, there's one question that's on the STAR test about tenacity.
Well,
Okay, so here's what the new
How
TEAS
many
guy
does it
says.
show?
Here's the boundary.
okay.
In Tier 1 instruction, students in biology are not expected to explore the differences between aerobic or anaerobic respiration.
Okay.
They're not expected to.
And I don't see how you can teach glycolysis, because glycolysis is an anaerobic process. And Krebs cycle electron transport, or electron transport is an aerobic process. I don't know how you can't tell them about that. And the internal processes within the cellular respiration, such as Krebs cycle or electron transport. So they do not have to understand Krebs cycle or electron transport, according to the Tier 1. Now, we gotta realize, when they say boundary for the Tier 1, that means the bare
Yeah,
minimum.
that's the
This is
bottom.
the bare minimum. If you want your kids to really understand it, yeah, you're gonna go into Krebs cycle, yes, you're gonna go into electron transport.
They don't take that long to teach anyway.
It doesn't, no, not from what we want them to know, no,
We keep it very simple.
no.
It's not that hard anyway.
In AP biology, my kids learn all the intermediate steps in Krebs cycle, and they learn all the intermediate compounds, and then we learn electron transport, we do the, but that's beyond the scope of what our kids need to have for this class.
No, it's like-
So I think the big concepts, big ideas for photosynthesis and cell respiration. Okay, so if we put our eggs in vinegar on Friday,
I think the fermentation lab is a good idea on the 19th, as an introduction to,
As opposed to the gummy bear lab.
Yeah, maybe.
Okay, alright, fermentation lab.
and it'll also be fun, it's a good
Yeah.
lab to do on a Friday.
okay,
And then 22nd, we can start on photosynthesis, oh my god, this is
Well,
nice.
no, we've got
Our ATP,
ATP first.
so background info.
Pardon the interruption, students. I need Noah Doty and Savannah Jennings to the front office, please. Noah Doty, Savannah Jennings, Axton Mendez, Sophia Rodriguez, Amari Williams, Donovan Cato, Nina Sanchez, Zari Johnson to the front office, please. Zari Johnson, Sophia Rodriguez, Amari Williams, Donovan Cato, Nina Sanchez, Axton Mendez, Savannah Jennings, and Noah Doty. Please come to the front office. Thank you.
So how long are those notes?
Background information, that's on
It's just
page
one
one,
page.
it's just one page.
No, when we say one page, like.
And then
Sorry,
there's,
this one.
and then there's the page, the other page has the background on the biomolecules, doesn't it review
Yep,
biomolecules?
carbohydrates, lipids, proteins, and then ATP, ADP.
So there's our spiral.
And
Yeah.
then our glycolysis, well, the background information, and then we've got a little bit of notes on fermentation.
Make that connection.
That's 1 day for photosynthesis and 1 day for cellular respiration.
Yep,
Cellular, like,
and then we're gonna.
photosynthesis or whatever, that's not that hard to get through in 1 day, but cellular respiration is,
Well, if they don't have to know what's going on in the Krebs cycle, they don't have to know what's going on in
So,
the most they need to know is that, you know,
The equation?
they get the equation, they get 2 ATP from glycolysis, they get 2 ATP from Krebs cycle, they're gonna get 34, they're gonna get 20, 34, 32, 34 ATP from electron transport. I think if they know that and they understand that electron transport is where you get your biggest bang for your buck, which is, I try to always tie that to, you know, the final electron acceptor in cell respiration is oxygen.
1 day's not enough time.
I don't think, I think you're right.
Well,
So that would be Tuesday,
photosynthesis.
photosynthesis.
Yeah, we're gonna have to smash,
And
'cause
cellular
I like,
respiration.
we can't stop, so Tuesday's gonna have to be photosynthesis as much as you can get through and as much as you get through for cellular respiration.
Which,
But, I mean, photosynthesis is still a lot, right?
it is a lot,
Yes.
agreed. Alright, so then we've got the relationship between the two, and then on a cellular level questions.
And we're out of time though.
Yeah, well, we could do as part of the.
We could do
review.
that at the beginning of the class on
Yeah.
Thursday.
So it's
and
like a
then-
warm-up?
And then
Yeah.
as your warm up, tied it all together.
Yep,
Is that what it's called? It was,
The relationship.
relationship.
mhm.
And that's where they're gonna get that's where we want them to really understand it.
And then just hit them over the head with it 15 different ways, they'll get it. Some of it'll stick.
Alright,
So cell respiration, and then, if you have time, which I guess you probably will, we can do the booklet at the end of the class.
I usually do the book 3 times.
you can do it once.
Well,
You
I
can do it
give
once.
them 7 minutes, cause
Oh,
what I like
And I load it up in my Schoology folder
Yeah.
and let them.
Well, they also have study guides they could complete at the house.
Yeah.
We just may not have time for like the enzyme review. They can do it at the house too. And the
Enzymes
review, there's 3
is-
Well,
reviews.
and then we also got the
Enzymes
quizzes as
is-
well.
Oh, shoot.
Well, I mean, we can,
We've got 3 quizzes in this unit.
yeah.
So,
We gotta figure out how to fit.
I'm gonna probably tell them, the 18th? Tell them to do the quiz
Homeostasis
for
one?
homework.
Yeah, I'll give the
So this is
homeostasis
quiz
one.
1,
Yeah,
Mhm.
that's the homeostasis one is the first one.
okay.
The second one,
Where will we fit the second one in?
let me see what the second one is.
Good question. Cell energy quiz 1 and cell energy quiz 2.
Let me see what this is.
Pardon the interruption, students. I need Savannah Jennings to the front office, please. Savannah Jennings, please come to the front office. Thank you.
It
Quiz,
has both photosynthesis and cellular respiration on it,
on which one, the first quiz?
on the first one.
And the second one has that as well.
So maybe we could just give one, because
Just give one.
We
honestly,
can give it on Wednesdays like
Wednesday.
it's like homework, I guess.
with all their 3 study guides.
I mean, to be fair, if we wanna hit them over the head 15 different ways, this is how we do it.
Okay, well, if we finish early enough cellular respiration, maybe we could give it in class.
I
And
don't
do
think
you
I'm
guys
gonna
wanna
finish.
do quiz 1 or quiz 2, cause I can.
What's
I
the
say-
difference?
I say assign both of them and just give them to them homework, cause it's gonna be a good review for them.
Yeah, just make
Because
it on Friday.
because some of those questions they just may see on the progress check,
So Wednesday night, give for homework.
and tell them it's due Friday.
That's what,
Do
that's a good
Friday.
idea. That's a good idea.
Okay.
And give them what, 2 chances or just one?
I'm good with that.
2?
I mean, 'cause they
Give
can-
them 2 and just take the average of the 2, because
Alright.
if they're taking it at home, they can Google the answers, they got their
Yeah.
notes.
That's
And,
true.
and then they don't deserve the higher.
So how many
No one's
different,
up in the higher the 2 kids.
And I think as
I do
we're
it for the
going
child
through,
section.
Yeah, I do that too.
as you're teaching,
But as far as the
um,
they're doing the average.
as you're teaching active transport,
Mm-hmm.
you need to make sure you hit the function of those proteins, moving stuff in and out.
Proteins, yeah.
As you teach passive transport, yeah, hit that. Also facilitated diffusion, that's one that I always like to test. Hit that again, the transport protein. So we're spiraling back in unit 1, and getting some biomolecules in there. And then of course, when you do photosynthesis and cell respiration, you're gonna get carbohydrates again, and you're gonna hit enzymes again, because they need to understand that the reason photosynthesis happens is because of the enzymes that plants have to do photosynthesis.
yeah,
Where would you go to find those enzymes? I always ask my kids, where would you go to find the enzymes to do photosynthesis?
and then you get to spiral back in,
And then you get to spiral back in the organelle and
Organelles, yeah.
chloroplast. And then I ask my kids, where are you gonna find the enzymes to do cell respiration? And they're gonna spiral back in mitochondria.
Yeah.
Cause I want them to think about that. And then we get to AP Bio, in AP Bio, we talk about the differences in the chemistry and the differences in the pH. There's a big difference in the pH inside the mitochondria, inner membrane versus outside the membrane, cause it creates a proton gradient and
Yeah.
much higher, much more acidic on the inside. But we can spiral back in biomolecules through this, which we need to. That's one we need to spiral back in,
For
because they're
some
gonna see
reason,
that.
it's not registering in my brain.
And then enzymes, we'll hit again when we get to DNA replication.
I like this is way too much to squeeze in 2 weeks, but
It's
we'll
not-
make it work.
Yeah, I
What's
understand.
the question?
Tonicity, that word is not registering in my brain.
Oh.
Tonicity, hypertonic, isotonic.
Okay, now, I got it now.
You
Don't you
said
remember
what
biology
to
degree,
really, so photosynthesis
Uh,
enzymes, and when we get to the
Yep, since
protein, right?
19
When we get to
92.
protein
Born in
synthesis again,
1993.
I'm sorry, when we
So,
get to DNA,
yes,
when we
we
get to
still had,
rolling
We did
nucleic
like-
acids and, the next unit, which would be,
Okay.
We didn't have
the
a
rolling
chemistry.
nucleic acids. When we talk about DNA replication, we'll hit
Except for
enzymes again,
DNA
because they have to learn the enzymes that are involved in DNA replication. So we're constantly trying to spiral back in that unit 1 material from biomolecules,
Alright.
and then link it to the cell again.
We need a bridge day on the 29th. I don't care what they say, because these kids are gonna miss some questions.
29th bridge.
Oh, yeah. Alright, but then what are we gonna do? Are we gonna try
notebooks.
set up notebooks for the next unit?
But we said,
Like,
That benchmark has to get, there's no way we can do this with that benchmark.
Let me look and see.
okay,
So
what did you say the benchmark is supposed to cover?
how does she expect us to cover mitosis in 6
She's not a biology teacher, that's how she expects it.
She's not looking at that, she's not in the classroom, so people that are making these calendars are in the classroom, and they used to use teacher input,
And
and
then,
they quit doing that because apparently somebody downtown knows more than we know
Oh,
about
God.
teaching biology. So 6A, B, and C, so let's see what that is, 6A, B,
okay,
and C.
so we got cell cycle, we got mitosis, we got DNA structure, are we still gonna do DNA structure?
6A, explain the importance of the cell cycle, growth of organisms and stage of the cell cycle, including DNA replication. 6B, explain the process of cell specialization through differentiation, including the roles of environmental factors. And then C, relate disruptions in the cell cycle to diseases such as cancer. So really, it's just cell
Mitosis,
cycle,
differentiation.
mitosis, cell differentiation, and then cancer or apoptosis. That's where we get into apoptosis once again.
Right.
No, that's
Are
a
you
5
gonna be
minute
able to get us
conversation.
gummy bears?
Huh?
Oh, we're not doing gummy bear lab,
Never
remember?
mind, okay.
I don't think we're gonna have I don't think we have the time.
Where's the
Well, see,
space?
cause like me, cause I did active transport yesterday, tomorrow we'll be modeling it and just reinforcing that if something's too big to come through, you've got to have a way for it to get through.
I was gonna do, my plan was doing, if I can get it printed today, is I'm gonna give 6 kids a diagram with a phospholipid. They're gonna stand opposite each other. And in the middle, I'm gonna put a kid in the middle with a picture of a protein, transport protein. And then I'm gonna have kids come up and they're gonna either slide through the membrane, like oxygen would slide through the membrane, carbon dioxide would slide through the membrane, and then I'm gonna have a molecule like sugar, and then sugar's gonna bounce off the membrane until it hits that transport protein, and then that transport protein's gonna grab it and pull it across. I was trying to figure out a way to get the kids up and to kind of visualize this so they can see it. Because it's such an abstract kind of a thing right now for them.
It's not a bad idea.
You know, you could do 8 kids. 8 kids with the phospholipids, and then put a transport protein in the middle, and then have a couple of other kids come up and one hold carbon dioxide, one hold oxygen, and one hold glucose. And then the carbon dioxide and the glucose can just, or the carbon dioxide and oxygen can just slide through the membranes. But the sugar wouldn't be able to slide through the membrane, way too big.
Well, could you have kids like join hands and these guys are a sugar molecule?
Yeah, you could. Yeah, there's a
That
lot of
way-
different
that way
ways.
it's
I like
more
that one.
visual.
I like that one, cause the difference between one kid can fit through, like
C6H.
2
So
kids.
you could have a
can't.
carbon, a hydrogen, and an oxygen.
Yeah,
You could have a
yeah,
kid
yeah.
who's a carbon, a hydrogen, and an oxygen, and make
They
them.
could be by themselves and make the
Yeah,
sugar.
that's a good
And
idea.
you could do
Because
one kid for
it's
carbon, 2
technically-
kids for hydrogen,
Alright,
one
alright,
kid
alright.
for sugar, for
Alright,
oxygen,
That's a little bit better.
and then you got your 1 to 2
Why
to
don't
1
I just do
ratio.
6 kids for carbon and then we?
Yeah,
And that way you can, because I was just trying to think to myself, how can I get
We
them
have
to
so many
visualize
kids
this?
in
Yeah,
the
8
class.
and then 12 for hydrogen. Why not?
Because when I do cell growth, and I show them why cells can't get to be too big, I take my kids outside, we form a circle. I start off with a circle that's just, I start off a circle
I do
with
it all
just,
the time.
with 2 kids, and
Make it
then,
bigger.
and then I get, make it bigger and bigger, and then I carry a bag of sugar around, and I demonstrate how the sugar would cross the membrane, and then the ones in the middle never get the sugar,
Yeah.
so.
You can do it with a string too. I remember doing that in college.
Area fire is gonna be used.
So something to that effect. So here's my thought. I think this is gonna be a stretch because of this. I'm gonna shoot for, in my classes, I'm shooting for 85% on this progress check.
Oh, that was nice.
Yeah, no kidding.
Well, I gotta remember.
Well, that's my goal. That's a goal, 85%.
Well, yours will get that.
For progress check number 1 for this unit. That gives us until the 25th to review. Progress check on the 26th. I am gonna email Sandra here in just a moment, and I'm gonna ask her,
Not taking mitosis.
I don't know, she needs to, I'm gonna suggest she check with other teachers to see if everybody is on the timeline,
Can we email her too?
to make
We're
sure
behind.
that we get to the 26th,
Yeah.
because it's gonna be a push, for us to
I
get to the 26th.
think this is fine though. Like this is a lot, but this is doable.
Okay,
It's doable.
it is, but,
It's not ideal.
yeah, exactly. Like, we're supposed to be spending time doing labs, and we've got,
We can't do labs.
Yeah,
can
there's
do
just
labs.
no time.
We got 2 labs.
There's literally no
Really
time.
sad.
no, the
Put it in
kids
that.
aren't
So we're gonna
physically,
get a lab
the
on
exception
Friday.
weren't doing any labs.
Yeah,
We'll
I
get
get
fermentation lab on
I feel bad
Friday.
for the kids.
School of Jesus went down.
Yeah, the internet's been going down.
They said the internet was spotty.
Well, internet is fine. It's just Schoology.
Bridge Day on Monday, anything, are we gonna do Bridge Day and that's all we're doing on Monday?
We can set up notebooks.
Yeah, we only go through the top 5 missed questions, but that's if we can
Well, no, no, no.
access the
No, no, no.
questions.
Look,
Can we
on
even
bridge,
access
on
it?
that bridge day,
Yeah,
after we
we should be
discuss
able
all
to.
that, let's separate the one picture that we have of the cell cycle.
And
And
let
just
'em
put that
color
on
it.
it and let them color it and fill it all
There
in.
you go.
Okay.
Because there's no discussion that you have to do. Because we can go ahead and get that colored and then we can
Good idea.
actually take class time
That's a good idea.
to discuss it.
Cause we should be able to see if they progress check on Friday, and you're gonna have some kids that are gonna need a progress check on Monday.
Yep,
But you
so we
should,
can't really do bridge day.
I mean, there's always gonna be kids that
Well,
miss it.
bridge
Well,
day,
is she
we're-
even gonna have it open for us?
yeah, it'll be there, it should be accessible. I don't see it, she told me last year she wasn't gonna close anything,
And then she closed
and then
everything?
she closed everything, but this year she said she's gonna leave everything open, so we still have access to it. So if she leaves it open, we still have access to it, that shouldn't be an
Okay.
issue.
I mean, my concern, the reason I think we need the bridge day is because we're not 100% confident with this.
Okay, cell
This is the
cycle.
best we
I
got, so
feel
we
like
gotta
we-
help the kids.
I think
Be able to
our
reteach.
biggest concern is time, and
What is this next unit supposed to cover?
So we showed
Mitosis,
photosynthesis,
Just
cellular respiration.
DNA and DNA?
the next unit is,
and cell cycle.
Just cell cycle, which is DNA,
this should
DNA,
be cell
mitosis.
cycle.
right here.
Should be
It is,
6A, 6B, and 6C, so that should be cell cycle, that should be cell
We have
specialization and differentiation.
That's a
And then
differentiation.
the role of disruption of the cell cycle and how it leads to the development of
So,
cancer.
cell cycle, apoptosis,
cancer.
So no DNA
Differentiation.
structure?
DNA does not come in until, and I don't like that because.
Right.
We talk
have to-
about
We talk about
replication.
DNA replication, but we don't show how DNA replicates until we get to the role of nucleic acids in the next.
Hey guys, we just need you to know DNA replicates, don't worry about
So
it.
this is where you copy your DNA.
Yeah.
So you're just gonna
We've
tell them
always
this is where
done
you
it
copy.
together, DNA
Yeah.
and.
We usually
cell
do
cycle.
DNA and we usually include that.
It's DNA, that we even did it last year.
Yeah.
So
Yeah,
pull
we
up
did.
let's pull up our unit on, cell cycle and let's
That's
look and
right.
see what
Hang
we're
on,
doing.
hang on.
So we're gonna
Because we
start
gotta make
with,
sure that,
cell
Schoology's
cycle.
working.
Schoology's up again, so.
Yeah, I'm in Schoology.
You've got to talk about leading string, because you have to talk about what replication is. because
Yes.
Oh,
it-
God.
So you
Leading
have
string,
to talk
lagging string.
about,
Lagging strands.
So we're gonna have to hit cell cycle on that Monday.
Yeah.
That Tuesday, we can
and then
hit,
DNA structure is after that,
hit cell cycle.
because
And then
then we
related
explain
to-
how it happens.
Mitosis, do we go ahead and go into mitosis on,
I mean,
Tuesday?
yeah, it's kinda hard to talk about the cell cycle without talking about mitosis.
So if we're gonna
Yeah.
color it here, discuss it here, and then jump into mitosis on Tuesday?
Yeah, I think, yeah, we probably
Go,
won't finish that.
go through the cell cycle and then just jump into mitosis, but you won't finish it.
Yeah.
Right, so, yeah, so.
You may be able to do that
cell-
first page of mitosis.
Well, cause the
Okay.
first page of
Okay.
the cell cycle is this,
Right.
is that big
Right,
spiral.
right.
Yeah,
Right.
that.
What's this thing?
Coloring sheet.
But then
That's
we've
what we're gonna
got
have to do
this
on
part.
That's what I have to do that on Monday.
That's the,
Then
so
we
the cell
have-
cycle notes.
That,
And then, so can we think we can do both those in one day? If they color on Monday and then we discuss on Tuesday?
Yeah, I think
It's
so,
gonna have
I think so.
a lot of
Is
Okay,
the-
but do you
the
wanna do this part right here, the part where we have to,
heel stuff.
That will probably be, you think that's, I think that's Wednesday.
do we really need, okay, question, do we need to go in that depth, or do you want?
Well, the tier one boundary says the kids don't need to know the phases of the cell cycle.
That's what. I was
However,
asking.
you and I both know that on the EOC every year,
They
there's a
do it.
picture of a cell in interphase, or I'm sorry, a picture of a cell in metaphase,
Or
Yes.
or a
anaphase.
picture of a cell in anaphase.
Yeah.
So they're gonna need to be able to recognize it, even though the state,
Gotcha.
the teaks, it says that that's a tier one boundary.
I'm
It's-
okay
You
with
gotta
that.
teach beyond that tier one boundary.
It's not that hard anyway, there's like 4 major differences.
Yeah.
It's
you
fine.
start out with that, because this is what they'll be doing while we're doing the
Okay,
bridge test.
so where are they setting up the notebooks and all
We
that
actually have this
stuff?
picture.
To actually
Uh,
have
that's not
the
today. That
information
was
to put in
yesterday.
there.
That's what I was saying we should do on the 29th.
Wait a minute.
I set them up after.
That'd be good, I like
And
that.
then
It's like
they
this.
have
We should set up notebooks on the 29th,
Okay.
move everything over,
And then that was it. Then we talked about DNA structure.
because we don't have a notebook yet.
Yeah.
Some of this stuff is this unit, I feel like.
And then if you have time at the end, you can,
For college students.
I guess bridge, or you can start on the,
Well, that's
But
because
that-
they
Yeah,
planned
you,
it.
Yes.
you could bridge after
That's what
the
I'm
kids
saying.
That's
finish
because
the
that's how
test.
they are making us do it.
Cause how
We're
long is this
cramming
test
literally
gonna
6
be?
units into 2.
With the progress check,
Be
be just like the last one we did,
So
15
15
questions,
questions, but there's
more
a
like-
writing portion, right?
more or less.
Feel better.
And I had no clue how to use Writable.
Yes,
I don't
there
know how to
is.
use Writable, I mean, I've
Is
tried.
that that
but-
one app that we didn't like, then they AI'd it?
That's the one that they did at the beginning of the school
Yeah,
year.
they-
That one was weird.
We need to
Yeah.
get, and I've already asked
we can go talk
Courtney,
to some of the English teachers.
Courtney said she would come down and give us a,
They need to actually.
and Kelly White said she would get coverage for Courtney,
They don't
so she can come down and show
We
us how to
should
do
do
it.
by
that
hand.
tomorrow, so we don't have to do
They
this.
should write stuff by hand.
It's hard to grade things that they write by hand.
I know, but
I don't disagree with you, but it's outside the purview of a science class, and we gotta work.
like, I don't
So I
think
think
we
we're
got
gonna
right.
do notes and then let them color, and then.
I think coloring first is fine.
Okay, I've got to.
'Cause
Color
we-
first, or
Color.
or go through it, or,
They take their time coloring, if you give them a time limit, it's so helpful.
Yeah, that's true.
so what do you wanna
That's
do
what I
on
like.
Tuesday?
Well, I mean, you guys do what you want, we can.
I'm gonna let them color first, and I'm gonna talk through it at the same time.
Okay.
Ask questions.
That's what I was gonna do.
Okay.
And then,
Open houses tomorrow,
Yep.
Yeah, it's open house.
but, I won't be here.
have fun.
Oh, that's fun, good,
It's usually like
I'll
10
be in
kids or
Dallas
so.
because of Midlothian,
because of what?
Midlothian, and then I'll be
Oh.
right back on the next.
Yeah. And that'll
Okay,
probably
like-
I'm
be
gonna actually
everything to
set
do
up
on
my
Wednesday.
egg lab tomorrow.
Okay,
And you
I'm
wanna
just gonna
go-
put them in the vinegar, so on Friday, I can take the 2 out of the vinegar and go ahead and put in the syrup, and so they'll be all ready to discuss on
yeah.
Monday.
Talking about
Help you set that
the
up.
egg.
Uh-huh. So
The
it has
only, the only thing I'm probably gonna do is I'm probably gonna put my eggs in vinegar on Friday and then set them up with the kids on Monday so they can see me set them up. so they can watch them on Tuesday and Wednesday and Thursday.
They're constantly reinforced.
So they're constantly reinforced, and every day they're gonna have to come up and look at those eggs. And then on Friday, discuss what happened.
And it's a great last second review
Yeah.
right
yeah.
before the test.
But yeah, it's gonna take 3 days for them.
So if we set them up on Monday, Tuesday, Wednesday, Thursday,
Well, two of them.
We're
can be
reviewing
set
it.
tomorrow.
Two
of them.
It takes
Yeah.
3 days
It's about
for the.
osmosis.
We set that up.
well, if we put them in there on Friday,
This Friday.
Friday, Saturday, Sunday,
We put them in vinegar to.
so Monday they should be ready
And then Monday
to
we
go into the different solutions.
And you'll see a change. Yeah.
It's cool.
Thursday, do you wanna go first? I mean, when you say stages of mitosis, you need the page that we pull back and write the stage.
I don't know if will we get to that point?
Clarity, there's no one here.
I thought somebody knocked, sorry.
Just here.
Gosh, them kicking
You
it.
know. Okay, so, is the color discuss
Sorry, I
is
thought I heard
that wheel page, and then the stages of mitosis is just that one pager, right?
Yes.
And then that means Thursday, we could go more in depth with each stage,
Alright, so we're not gonna print our.
because we don't need to go over DNA, right? So.
Not yet, we don't have to hit DNA yet.
So, yeah, Thursday we can go more in depth with each phase,
Yeah, well,
like the.
So
pages
stages
you
of
put
mitosis,
yeah.
what else? Cell differentiation, so
Yeah, I need
I'm gonna say
mitosis.
the benchmark, we got a review here.
Okay.
Gosh, I'm so far behind in writing this all down.
I'm gonna take a picture of it.
So that gives us mitosis wrap up.
Yeah. And then we can move into differentiation there too.
Cancer, cell differentiation.
We can also do like the amoeba sisters that day. 'Cause they
We
go-
got a quiz for that unit? Sure we do.
I'm sure we do. Yeah, we could do quiz.
Yeah, we do have a quiz for that unit.
Do that on Friday.
There's a vocab quiz and a
And a-
mitosis cell division
yeah.
quiz.
Can we do that on Friday?
Yeah, that's
Yeah.
fine.
So we're not doing the secret of photo.
Well, not yet.
Well, the vocab will also have DNA.
We can do secret of photo 51,
What's that?
right here.
It's a video. It's very
Why
long
don't we do
video.
secret
21st.
of photo 51 on the 9th?
Or we can watch it on the night.
And there'll be a lot of kids who aren't here.
Well, you're gonna have some retesters here probably.
Oh, yeah, yeah,
Let's
yeah.
do-
You could do it on Friday.
Yeah. Secret of photo.
Secret of photo
It's
51.
basically how Rosalind Franklin was the one who actually came up with,
She took the picture.
The
of
picture.
DNA.
and it's
Oh,
how.
that she got
men-
like.
I see her idea stolen.
Yeah.
They stole her
Okay.
photograph and they used it.
Man, that
See, I
one.
won't be here the 23rd or 24th.
You're just skipping all the.
Okay, so we've talked about,
Oh,
we've
Okay.
talked about the big teaks that that our kids need to hit.
Well, the
which is
Monday, that could be an
transport.
extension day.
Yeah, it could be, yep.
That
We
could
talked about
be-
the big teaks that we need to hit for these
Or
actually
we
3
can
units.
hit, we can go hit the things that our kids, like, we can look at the quiz over the weekend and see
Okay.
what our kids need a little bit more help with, and then extend after that for Monday.
The only thing I wanna do is, and I've already got a PowerPoint started. I pulled all those questions from last year's EOC
Mhm.
that were related to
Can we do
photosynthesis and
that
cell
on the 6th?
respiration. And I pulled the ones for photosynthesis and respiration, I pulled the ones for, cell transport, and I put all those on a PowerPoint. So I've got a PowerPoint, and we can do that on the 6th,
Okay.
if you want to.
Yeah.
And that'd be a great review those old EOC questions.
Yeah,
And it'd be a good review before we look it, and then take that benchmark and see how we do on the benchmark. Again, I think we need to have a goal
a
here.
study guide.
What's our goal for the benchmark? Where do we want our kids to be?
I mean, just to be
100%.
clear, we hit everything,
I would,
and then we
I
got
would love to
an
see
extra
my
day to
kids
actually
do
go a little
something
bit more in depth,
for my inclusion.
right?
Right.
So,
Where do we want our kids at?
I mean, your honors
Well-
kids probably hit a 90, right?
I'm hoping.
Yeah, honors, we expect 90, regulars, we could expect
I'm
what,
shooting for an 80,
80?
yeah, I have some
Honors
kids that are gonna,
inclusion,
some are Spartan kids,
Love it.
so.
some of our, yeah,
And the 9th, what about, that's gonna be finished up.
finished exam. Can we also set up notebooks for the next unit that day?
Yeah, that's fine,
Are they, is there gonna be a writing portion for that one?
I'm sure.
For the
what does
benchmark, yes.
EXT mean?
I think that,
Station.
I believe the benchmark, I have to go back and look to be sure, but I think the progress checks and the benchmarks all have short constructed responses.
Okay.
So I have to go back
Does
and
that make
double check.
sense, old lady?
I have to go back and check, but I
I'm just,
believe it.
My brain's just not going as fast.
I believe the benchmark and the progress check will have short constructed responses.
I mean, that's what they said they wanted them to, because the EOC.
And I'm interested to see how that's gonna be graded. Because if we have to sit down and grade,
Come on, we know how this, copy, paste, AI,
yeah.
grade this one.
Oh, yeah, absolutely.
So we made some SMART goals for our kids, for our progress check and our, benchmark. We talked about the big EOC or the big TEKs, for these 2 units, and we've actually squeezed in homeostasis, we've been in photosynthesis and cell respiration, we've put processes for homeostasis, and then we squeezed in, unit 4, which is mitosis.
Do you wanna also put in a vocab quiz somewhere in there too?
You can do it for homework.
Yes, because there is a vocab in there, one for both
There
of these.
is, and I'm looking at it, and it has things like,
Has DNA
double
stuff.
helix and DNA, but those were the only 2, and chromatin, but they should know chromatin.
Well, that's what I was thinking for
But
the
other than
Very
that,
chromatin.
they should be able to get.
So is that vocabulary for cell respiration photosynthesis, or is that vocabulary for
Cell
cell
cycle
cycle?
and DNA.
Is there a vocabulary for photosynthesis and cell respiration also?
No,
What?
no, there's no vocab.
Okay, I thought I put one in there.
We really don't have time for
You
one.
did one, but you didn't
Oh, I think I put it in there.
No, you just didn't put it in the group, cause
Ask
it's
that
something you
question
created
again,
that
Mark.
one.
Cause
What
I
did you
would have had it.
What's that?
What was the question you just asked?
The photosynthesis and cell respiration vocabulary quiz.
No.
Is there one in the
No.
unit there?
No,
No, there is one
Maybe
for
we
cell
didn't.
cycle.
Okay, maybe we didn't do that for photosynthesis and
we
cell
just
respiration.
really don't have.
We probably didn't do it last year because we were so jammed up.
Oh, that's
We were
right.
so jammed up with time.
Well, we
Again.
can just check the previous year, see if
Again,
it's
I'm gonna try
I
to
have a
push,
bunch of investigations in here we could be doing.
I'm gonna try to push Sandra to move. I don't know, it's just
But can we make it through
We-
mitosis without the DNA?
we can make it through mitosis without the
I'm
DNA.
telling you, that Monday,
Okay.
for that extension, we can add in DNA there. For the kids who need that extra little bit, or that we're confident can go a little bit further,
That's
that's
your
the perfect day
extension.
to add it.
That's your extension.
They just need to know that when DNA replicates, just making a copy of itself.
Yeah,
Because when we
That's
start
it.
to come back, we'll hit DNA, we'll hit leading strand, lagging strand.
So
The
maybe
enzymes involved.
those kids who, that extension will help them a little bit more on the bench, maybe we can push them to 100, maybe.
Okay, so we okay with this so far?
I like it so far. It's still too much.
I would like to go ahead
Too
and
much.
plan I would really like for us to, in the next next hour or so, let's go ahead and let's flesh the rest of this out, make sure we got it where we want it. But go ahead and jump into next 9 weeks
Yeah.
and look at what we plan and what we think our calendar would look like
okay.
and compare it to what the district's calendar is, and we can maybe make some suggestions, to Sandra, the DIS, and say, hey, we really need to think, do we need really 31 days for us to do a whole 9 weeks? Because
Right.
you and I know, we can teach the role of nucleic acids,
Mhm.
and you can teach gene expression 10 days
Yeah.
instead of 31 days.
Mhm.
So if we can do it in 10 days, I think it'd be much, I think we need to just make some adjustments there. But I think we can do that.
I agree.
He doesn't need us.
Hmm?
He doesn't need us. I just saw the shadow behind. Are you near?
Let me check my
Okay.
equipment here.
Here's,
I think it was a student. Or was it First Sergeant Baldwin?